Hello everyone, my name is AJ Keefe, and in this presentation I'll be reviewing the pathway of amyloid beta production and also axonal lysosome transport. I'll take these two concepts and integrate them by covering this paper titled Impaired JIP3-dependent axonal lysosome transport promotes amyloid plaque pathology, published in the Journal of Cell Biology. I recommend that my viewers pause each slide and read the material and analyze the graphics before playing my commentary. And if you have any questions, please comment below and I'll promptly respond. Thanks for watching. Amyloid beta is the principal component of the amyloid plaques that are observed in Alzheimer's disease and are actually composed of short peptides derived from a protein called APP. APP stands for amyloid precursor protein, and that's about all that's really known about this protein. Its function is quite mysterious. So these amyloid beta plaques that suffuse the diseased Alzheimer's brain are chock full of these peptide fragments derived from the APP protein right here. Since APP, the protein itself, is completely harmless, researchers have been especially curious about what factors lead to the proteolysis of APP that ultimately produces these toxic fragments, leading to aggregation. So APP cleavage at position beta by a protein called base, and cleavage at position gamma by a protein called PSIN 1 or 2, produces this critical 42 amino acid amyloid beta peptide that we see aggregating. In this graphic to the right, we see that full-length APP, right here, full-length APP contains 3 very critical cleavage sites huddled around this APP's its transmembrane domain right here. So there's an extracellular alpha cleavage domain right here, and also a extracellular beta cleavage site that produces a longer fragment than the alpha cleavage site. And there's also a transmembrane cleavage site that's recognized by the gamma secretase, or presenilin 1 and 2. When APP is cleaved at the beta and the gamma site, but not at the alpha and the gamma site, it produces this peptide called amyloid beta 42 that contains this beta strand that can link up with other amyloid beta strands and becomes incredibly stable. Multiple amyloid beta 42 peptides can form these insoluble beta plated sheets that are highly resistant to degradation, even in lysosomes. And these amyloid beta peptides can also be found at a length of 38 and 40. But amyloid beta 42 has the greatest propensity to aggregate. And interestingly, the amyloid beta species that are shorter than 38 are non-pathological. And you can see that the alpha cleavage site right here, this alpha cleavage site, produces a shorter amyloid beta fragment. And it's actually short enough as to where it doesn't produce pathology, and it does not aggregate, it doesn't cause disease. So an alpha cleavage is a very good thing. So the generation of the pathological amyloid beta 42 fragment revolves around the interaction of these 3 main proteins: APP, the protein itself, base, right here, and presenilin 1 or 2, or PSIN 1 or 2. APP is again a protein of unknown function that is largely localized to the synapse and is constitutively shuttled through the ER, the Golgi, plasma membrane, and then is degraded in lysosomes. APP is also a highly evolutionarily conserved protein that has been observed aggregating in other primate species and has some role in synapse formation and repair. So APP, the improper cleavage of APP can lead to aggregation in other species like monkeys as well. So, and it also has some role in synapse formation and repair because the loss of APP results in the destabilization of synapses, although it's not very significant because animals lacking APP still survive just fine. They just have a very slight memory deficit, but it's nothing extreme. It's nothing like Alzheimer's or dementia. The problem with APP is not exactly in the protein itself, but it's how the protein gets degraded. So base is the protease responsible for the cleavage of APP at the beta site, which is very bad news. The pathological implications of this cleavage at the beta site are implicated or illustrated in individuals that lack the critical amino acid at base's APP cleavage site. So when APP's beta cleavage site is mutated, these people have 40% less amyloid beta accumulation in the brain, and they almost never get Alzheimer's disease. So the presence, or I guess the cleavage at the alpha site, which doesn't produce pathological fragments, is very much preferred. And when the beta cleavage site is prevented by mutation, the people with these mutations almost never get Alzheimer's. So base tends to produce these harmful beta cleavages during protein sorting in the endosomes and the Golgi and the lysosomes. So base kind of has this tendency to function in the early endosomes and the lysosomes. So this process of base secretase followed by gamma cleavage and amyloid beta production is occurring largely in the endosomes and the lysosomes. So as a result of that, a lot of the amyloid beta fragments actually begin their process of aggregation in the endosome. However, to complete the production of the amyloid beta 42 plaque, APP also needs to be cleaved at this transmembrane gamma position. And this is done by the presenilins 1 or 2, which can imagine from their name, presenilin, they're implicated in senility and Alzheimer's disease and dementia. So PSEN 1 or 2 is simply a protease that cleaves APP as transmembrane domain, and there's various activating mutations in presenilin 1 or 2 that are responsible for early onset Alzheimer's disease. So by judging by all these various mutations, the obvious accumulation of amyloid beta 42 in the brains of Alzheimer's patients, it's quite clear that APP processing is fundamental to the pathology of Alzheimer's disease. But where does it begin? Well, it turns out the processing enzymes base and presenilin 1 are primarily localized to endosomes and the Golgi and the lysosomes. And so it's theorized that the initial amyloid production process begins in the early endosome following endocytosis from the plasma membrane. In this top right figure, we see the aggregation of amyloid beta following endocytosis. Since the protein can form stable beta sheets, it actually resists degradation in these very hostile high pH or low pH environment of the endosomes and the lysosomes. And when these insoluble aggregates accumulate in the lysosomes and the endosomes, they are often secreted or exocytosed, where they end up extracellularly and they promote the regional inflammation, and they actually disrupt synaptic signaling. And they can even bind a lot of different receptors and trigger pro-apoptotic gene expression in other cells. Another primary idea tackled by this current paper relates to how amyloid beta accumulation in endosomes and lysosomes disrupts trafficking. So remember that APP is primarily a synaptic protein. So when it's degraded in endosomes and aggregates, this often is occurring in distal axons in near synapses. And in a healthy normal axon, endosomes, as well as autophagosomes, are constantly turning over their contents. And for reasons that are not fully understood, this process of axon protein degradation is accompanied by retrograde transport through here. So endosomes or autophagosomes, I will be using these largely interchangeably, are loaded with substrate to be degraded, and then they are actually retrogradely transported towards the cell body by dynein. So as these organelles are trafficked, retrogradely transported towards the cell body by dynein, they are literally acidified, and they slowly morph into lysosomes, or sometimes they simply merge with a preformed somal lysosome. So you have this maturation process by which an early endosome or an axonally derived autophagosome acidifies into a late endosome and then a lysosome. And this process is critically marked by the acquisition of lysosomal enzymes and also by the switch from Rab5 to Rab7. So you see, as early endosomes and autophagosomes retrogradely transport, they mature into late endosomes, and then eventually they acidify into mature lysosomes, and or sometimes they simply just fuse with lysosomes. In this process of retrograde transport is marked by the switch from Rab5 to Rab7. So the concept of Rab proteins are actually a very important concept, because Rab is essentially what determines the identity and the destination of any given vesicle inside of a cell. So Rab proteins are simply a GTPase-like protein that when loaded or holding on to GTP will recruit various effectors that determine the functionality of that vesicle. So a vesicle holding on to Rab3, for example, is headed for the synapse. It's recruiting proteins that direct it to the synapse. A vesicle holding on to Rab18 is a lipid droplet, recruiting proteins related to lipid droplets. A vesicle holding on to Rab1 is in the Golgi complex, recruiting Golgi-associated proteins. But the central player to this paper is Rab5 and Rab7. Rab5-coated vesicles are early endosomes. And as these Rab5-coated vesicles, these early endosomes move from the synapse towards the cell body, they undergo this process of Rab conversion, where they swap Rab5 for Rab7. And Rab7 is a marker of late endosomes and is actually required for lysosomal maturation or fusion. Notably, a Rab7 effector, so a protein that binds to Rab7 GTP, is JIP3, JIP3. JIP3 is a Rab7 effector that bridges late endosomes and autophagosomes with dynein in axons for retrograde trafficking. So think of JIP3 as a scaffolding protein that allows Rab7-coated vesicles to link up with dynein so they can move retrogradely. And the loss of JIP3 selectively disrupts axon retrograde trafficking of Rab7-coated vesicles, but it does not affect the dendritic trafficking or the somal trafficking of lysosomes or late endosomes. So if a researcher wanted to selectively disrupt axon retrograde transport of late endosomes, they would knock out JIP3. And this leaves intact the transport of things like mitochondria, synaptic vesicles, everything that's traveling anterogradely, dendritic transport, etc. It's a very selective mechanism to disrupt late endosome retrograde trafficking. So if a research team wanted to see how specific defects in axonal retrograde endosome transport affected something like amyloid beta production, they would look at the effect of JIP3 knockouts. And that's exactly what the current paper did, is that they knock out JIP3 to essentially block this process of early endosome to late endosome to lysosome maturation by knocking out JIP3. And this prevents late endosomes from linking up with dynein, and so they're unable to get retrogradely transported and mature. So previous literature has found that JIP3 knockouts caused a loss of axonal retrograde endosome maturation and transport, resulting in axon swelling and cell death at 3 weeks of age. So moving endosomes back to the soma is a very critical process. We also know that a hallmark of amyloidogenic brains, so brains that produce the pathological 42 amino acid amyloid beta fragment, a hallmark of these brains are these swollen dystrophic axons with severe organelle accumulation. And I believe the impetus for this study was at least partially motivated by recognizing that both JIP3 knockouts and improper ATP processing both produce this similar phenotype of swollen axons. In Alzheimer's disease, these swollen axons are saturated with late endosomes and autolysosomes, which are basically a combination of an autophagosome and a lysosome. But both of these vesicles are chock full of amyloid beta. They're also full of its processing enzymes base and presenilin 1 and 2, as well as APP itself. So these dystrophic swollen axons that are observed in Alzheimer's disease are full of amyloid beta, APP, and its processing enzymes. Also, curiously, these stalled late endosomes and autophagosomes lack all lysosomal enzymes, meaning they are derived from the synapse and are failing to develop into full-fledged lysosomes. They get halfway there, then something goes wrong. There's a significant amount of research and literature that it suggests that improper endosome and lysosomal processing and production of amyloid beta is ultimately what's killing the neurons by preventing the maturation of these organelles into lysosomes and causing axon dysfunction. So in the current paper, the authors wanted to model retrograde transport and endosome malfunction in axons, and they took advantage of these JIP3 knockouts. And they examined the effect of JIP3 knockouts on amyloid beta production and other hallmarks of AD, or Alzheimer's disease. They began by first confirming that loss of JIP3 results in the characteristic swelling of axons and the accumulation of these autolysosomes and late endosomes. And additionally, the accumulated organelles lacked proteases, suggesting that they were coming from the synapse. Next, they observed that ATP processing enzymes were abundant in JIP3 knockout axon swellings, as well as amyloid beta 42 itself. So these JIP3 knockouts are actually showing the molecular signature of Alzheimer's disease. That right there I thought was really tremendous and amazing. JIP3 knockouts almost create a model of Alzheimer's disease. And so they took heterozygotic JIP3 knockouts that were haploinsufficient, so they only have one functional copy of JIP3, and they crossed these mice with an Alzheimer's disease model of mice, and they found that lacking one copy of JIP3 significantly increased the number of plaques and amyloid beta production. This ultimately suggests that JIP3-mediated lysosome maturation and transport promotes amyloid beta 42 degradation, and disruption of this process promotes amyloidosis. So in other words, retrograde axon trafficking and lysosome maturation is a very, very important aspect to the formation of amyloid beta 42 and its associated plaques. Since JIP3 knockouts caused embryonic lethality, they had to extract cortical neurons from embryonic mice before they croaked. And what they found is that in these neurons, they had swollen axons, as indicated by lysotracker staining. Lysotracker stains acidified organelles. And so the loss of JIP3 produces late endosome accumulation that mirrors axonal phenotype of APP overexpressing mouse neurons. We see the percent of neurons with lysosomal axon swellings is about 90% in the JIP3 knockouts and 0% in wild type. To better characterize these stalled acidic organelles, they stained for various lysosomal enzymes. They looked for cathepsin D right here, and they also looked at cathepsin B and AEP. These are all lysosomal enzymes, and what they found is that there were very low levels of each of these proteases in these axon clustered lysosomes that were in the swelled axons. But they were plentiful in the somal lysosomes. What this means is that these lysosomes that are stuck in the axon swellings right here are not derived from the cell body, because lysosomes in the cell body have adequate proteases and are functioning fine, and rather these organelles that lack proteases must originate from distal axons, locations such as from the synapse. And I should also note that GM130 is a Golgi-derived vesicle, and since there was no GM132 or 130 staining in the axonal lysosomes, it means that the lysosomes are not derived from the cell body. Otherwise, they would have this GM130 positive staining. And they also know that they're looking at axons because they have MAP2 stained. MAP2 is a marker of dendrites. And since there's no MAP2 staining right here, that means that this is an axon moving right through here. The authors wondered if these JIP3 knockout axons with accumulated autolysosomes also had high levels of ATP processing enzymes, like what's observed in Alzheimer's disease. And indeed, JIP3 knockouts had about 2.5 times more base in their neurons. And so this immunocytochemistry stands for both base in green, or I guess the image isn't green, and LAMP1. And they found that there was a lot more base in general, and a lot of it co-localized with the LAMP1, which LAMP1 is a marker of lysosomes. So we see a lot of base in these lysosomes. And since base is producing that harmful beta cleavage that leads to amyloid beta 42 fragments, this is probably not very good news for the neurons. Not only do these JIP3 knockout neurons have increased base, like we saw on the last slide, but they also have increased presenilin 2 in their lysosomes as well. And they also have increased amyloid beta production, almost 3 times more amyloid beta 42 in these neurons, which is simply amazing if you think about it. What the authors have done is they've essentially recreated the Alzheimer's disease phenotype in neurons by knocking out a lysosomal trafficking protein. Since JIP3 knockouts can promote amyloid beta 42 production by themselves, the authors decided to generate JIP3 heterozygotic knockouts that are haploinsufficient. And so these JIP3 heterozygotic knockouts only contain one copy of JIP3, but other than that, they live normally, they live to adulthood, they're fine. So they took these heterozygotic JIP3 knockouts and they crossed them with these 5X FAD mice. And this 5X FAD mouse contains 5 well-known Alzheimer's disease associated mutations in presenilin and APP. And what they found was that JIP3 haploinsufficiency significantly exacerbates the accumulation of amyloid beta 42. So these 5X FAD mice with only one copy of JIP3 had almost 3 times the amount of amyloid beta production, as we see right here. And this immunohistochemistry we're looking at is in the cortex of these animals. They're stained for amyloid beta in red and lysosomes in green. And we're seeing the accumulation of amyloid beta in haploinsufficient JIP3 mice. So these results suggest collectively that lysosomal trafficking is a central component to the production of amyloid beta and the subsequent formation of plaques. So our summary for this paper is that JIP3 knockout mice recapitulate many of the hallmarks of Alzheimer's disease, including axon swelling, immature lysosomes with ATP processing enzymes, and amyloid beta 42 production. JIP3 haploinsufficiency exacerbates the production of amyloid beta in a mouse model of Alzheimer's disease, suggesting that lysosome retrograde axon transport is a critical process to amyloidogenesis, or the production of amyloid beta 42. I hope you guys enjoyed this paper. Please comment any questions you have, and I'll be sure to respond. And thanks for watching.